Hi, I'm going to walk you through the electron transport chain process of cellular respiration. So we're going to start with glycolysis in the cytoplasm, and we're going to take, which includes taking glucose and breaking it down to 2-pyruvate, and with that we produce 2 ATP and 2 NADH. Those three products, 2-pyruvate, ATP, and NADH, are going to leave the cytoplasm and go into our matrix right here, and which is where acetyl coenzyme A activation will take place. So we have our 2-pyruvate, which we had produced before in glycolysis, and we're going to get a 2-carbon molecule, acetyl coenzyme A. And we're going to produce 2 of those, and doing that would also give us 2 carbon dioxide and 2 NADH. And when we bond 2-carbon acetyl coenzyme A with 4-carbon oxyl acetate, that gives us citric acid, which is a 6-carbon molecule. And so now we're also going to kick off coenzyme A, because it's no longer needed. And eventually citric acid will become oxaloacetate, and once that happens, we're going to have 6 NADH, 2 FADH, 4 carbon dioxide, and 2 ATP. And when we go to our electron transport chain is actually it actually deals with our electron carriers, so like NADH and FADH2, and we get 10 from our 6 NADH during the Krebs cycle, 2 from our acetyl coenzyme A activation, and 2 from glycolysis. And then our 2 FADH are coming from our Krebs cycle. And so these electrons are actually going to go through our electron transport chain as so, and they're going to come back into the matrix, and they're actually going to provide electron. Those electrons are going to bond with our half of an oxygen molecule along with our 2 hydrogen ions to give us a water molecule. So those electrons from our energy are going into our water molecule here, and also from the electrons going through our electron transport chain, those electrons are getting really excited, and that excitement is actually causing the hydrogen ions in the matrix to move against their concentration gradient into the intermembrane space, and so we're going to have more hydrogen ions up here. And so we started off with the isotonic solution, but now we have there was an isotonic solution between the matrix and the intermembrane space, but now we have a hypertonic solution in the intermembrane space and a hypotonic solution in the matrix. And this was actually due to one being forming water right here, so we're losing hydrogen ions to form water, and then the second one being we're pumping our hydrogen ions all the way up here against the concentration gradient, so we're losing even more hydrogen ions in our matrix. And so now this difference of concentrations will actually cause our hydrogen ions to go through ATP synthase, and they will go through like this, and ATP synthase with the hydrogen ions is going to cause it to have a spinning circular motion, and that spinning circular motion is actually going to turn ADP plus a phosphate into ATP. So now we have ATP right here from ATP synthase, the spinning motion. And in our electron transport chain alone, we're going to produce 28 ATP, but then for one glucose molecule, we're getting 32 ATP. This is because we're looking at it holistically, which is where we get 2 ATP from the Krebs cycle, and our other 2 ATP are coming from glycolysis. And so, yeah.